Before we start, right? Hi everybody. Welcome. If you're first time attending my class, you can say hi to me. Alright. So yeah, my name is Davin. For those who just came in, okay, let's go. Let's start. Okay, I briefly introduce a bit like our bio punya paper punya format lah, because maybe you all not familiar with it. Your teacher never teach you all before. Hello, what's up, everybody? Now, for bio, just like chemistry and physics, they are the paper two lah. Okay, I write here lah. Paper two. Paper two will have 3 sections in total. Now, section A is the so-called structured questions. So structured questions is literally what we've been doing all the time lah, means normal punya questions. Okay, there are 60 marks in total. And total got how many questions? 8 questions. Okay, got 8 questions. And the question punya marks, right, is, if not mistaken, is 55667788. Or is it 66778899? I can't remember. I think it's 55667788. So got 8 questions in total lah. Section B, we have is actually your essay lah, section B, and also section C. So we will call these two basically your essay lah. Okay. So for section B, we have 2 questions lah. So basically it's 2 choose 1 equals to 20 marks. Section C only got 1 question, compulsory must answer one, also 20 marks. So total your whole paper 2 is 100 marks. So I believe most of your punya upcoming exam, right, is the full format, means like 100 marks punya paper. So paper 2 is the main paper lah. Paper 1 is objective. Today we won't be doing lah. Okay, and I think most of your midterm won't have experiment paper, right? Okay, how many here got experiment paper, like paper 3 for your first term got? Got or not? For me, it's SPM format, means you all got the Amali, yeah. Okay, if you all got Amali, right, for the first term, right, I think most probably, right, it will be definitely be chapter 3. Only chapter 3 got experiment one. You can go to my YouTube. There's actually a few Amali, chapter 3 Amali punya paper that I got that you can watch over there lah. So unfortunately, Amali, I won't go through in this seminar. I will also go through in our normal class lah. Yeah, so okay, I'll show you my YouTube. Okay, so you come to wait my channel over here. Okay, so I think I got playlist one. You can find playlist. Okay, under here, my playlist here, right? You go and find, okay, this one, Selangor Amali 2025, got one chapter 3 one here. This is last year punya Form 4 and Form 5, their exam. This is a trial exam lah. Here got 2 Amali paper, 2 chapter 3 punya Amali paper, which is, one is the potato strip and one is the identify isotonic solution one. So for your first term, right, most likely is one of these 2 experiments. Or if it's not this one, right, it will be exactly like the same like last year punya SPM Amali. Wait, let me find it. Yeah, the one I didn't create playlist. That one you can look at this one. Is okay, I need to search. Is our Amali. Got that. Yeah, don't have. Public. Okay, got. You need to scroll down a bit. You look at this one. This one. SPM Bio Amali last year one. Yeah, this one also is chapter 3 one. So your first term, if got Amali, right, most likely will be this few experiment lah. So you can look at the chapter 3 Amali, last year punya Selangor Amali, or this last year 2025 punya SPM Amali. You just watch these 2 videos, should be more than enough already for your first term punya Amali lah. Okay, right. So we'll be focusing on mainly chapter 2 today. People who just joined us, we'll be doing chapter 2. Which chapter 2, yeah, we only have time to do chapter 2. Chapter 3, just like this morning punya session, I try to do, but I don't have time at all. So we mainly just be doing Form 4 chapter 2, which your first term, right? Chapter 2 definitely will ask a lot. Okay, now this chapter, actually quite important one. Even though it's like one of your first chapter, even in your SPM, your actual SPM, right, they will actually also quite to ask this chapter a lot. And this chapter is actually your bio punya basic. Means next time when you all learn a lot of bio things like what mitosis, meiosis that we learned recently, also got chapter 2 punya knowledge. Yeah, basically your bio will use a lot of chapter 2 stuff. Amali, how many marks? Amali is 15 marks only. That's why usually Amali, right, is not like the one you should be worrying of. The one you should be worrying of is your paper 2, which is 100 marks. So yeah, again, if those got Amali for upcoming exam 1, you all can go my YouTube and find a few videos to watch lah. Okay, so chapter 2, what we are going to go through today, okay, the most basic thing you all need to know is the organelles, which is also known as cell structure lah. Now, if you got follow my YouTube channel, right, recently I got do a video about organelle punya questions. If don't have, never mind, later we do also. And then other than that, there's this thing called the what tissue or cell organization. If you don't know what is that, that is basically, you know, your cell, tissue, organ, that thing. And then usually they like to test you on different type of tissues in human. We'll be doing that later. And then the last one also we'll be doing is this thing called the what unicellular organisms. Living process is unicellular organism, which is, you know, your amoeba paramecium. So today we'll be covering this. Can? All right. Without further ado, without wasting time, let's go. All right, so epithelial connective, this one exam got a lot. Usually they like to ask epithelial and muscle more lah, so we'll be doing those. If a lot, so not say a lot lah, but they will still ask lah, so we'll be doing that later. Okay, yeah, so by the way, people who just joined us, this set of questions that I'll be using, right, is different from the one in the website, because after that I did a separate one which include more like trial and exam punya questions. If you came in late, I will put these questions in the everyone chat there. You can go and download. Yeah, post it the second time there already. So if you don't have these notes, you just write down only lah. So you please just write along with me. So because if you just stare at the screen, you cannot learn one. Okay, let's go. Now, question 1, label organelles. So W. Everybody recognize what is this or not? So this one I like to call it like the Wi-Fi. The Wi-Fi ship thingy, that one, what is it called? This is your Golgi. Yeah, I believe a lot of you know, right? Correct. Is the Golgi apparatus. So if you watch my recent YouTube video, I actually got to tell you Golgi and smooth endoplasmic reticulum, how to recognize, right? Because some of you still confused with this. Okay, now, if you look at X this thing, right, is the layer thingy right above your nucleus. So this layer thingy that's sticking on top of your nucleus, right, is definitely your endoplasmic reticulum. Then a lot of this BG one, a lot of ribosome on top of it, that one is called the rough law. Like a lot of pimple, then your face will be rough law. This one, a lot of ribosome on top of it one. Yeah, so this fella is rough endoplasmic reticulum. So if it's smooth, right? Smooth and Golgi, right? The difference is, right? Okay, like I just said, smooth, usually the ER, right, they'll always be on top of the nucleus. You can see Golgi is further away from the nucleus one. Number 2, right? Endoplasmic reticulum, right? They are always attached to each other, each layers. Golgi is separate one. Like Wi-Fi, like we mentioned just now. Okay, and remember, let's write the full name. You cannot just write ER, understand? Okay, next, Y. Y is nucleus. This one's very simple. You can straight away. Next, give an example of a human cell that has high density of cell component Z. Now, they always like to ask this kind of question. What kind of human cell got a lot of what kind of cell component. So cell component Z is your, everybody can recognize this further, right? I think I can straight away mention mitochondria, you know, Okay, first, still remember what's the function of mitochondria or not? Okay, before we answer this one, okay, let's make sure everybody's on the same page first. Yeah, Sri, you're correct, the answer. But okay, let's mention mitochondria. Mitochondria punya function mainly is to produce energy, right? We always say we generate energy, right? Yes, correct. ATP in the form of ATP. What's the white ball? You mean this white ball? This one? Okay, that one is the outside the nucleus. Outside the nucleus got white ball. Oh, this one. This thing is this one. Or this one. That one is called the lysosome. Lysosome is like a sphere shape, but it's way smaller than the nucleus lah. Okay. Yeah, later we will do questions about that. Now, yeah, generate energy in the form of ATP. Now, today we're not going to talk about what's ATP, but ATP basically is like a chemical that provides energy for living things. It stands for adenosine triphosphate. Now, when they ask you what cell have a lot of mitochondria, meaning cells that need a lot of energy. So different cells in your body, right, need different cell component to function. If I give you an example, different shops that you go also need different things to operate. Let's say if you go to Mama shop, right, Mama shop will store a lot of flour, what else? Flour, Maggi mee, eggs, and so on, right? Because you want to make roti canai, make Maggi goreng. If you come to TTC tuition center, right, we will store a lot of paper, right? Because we want to use paper to print notes, for example. So same, what cells need a lot of energy? Okay, I saw you a lot already spam me the answer already. Very good. The most common one is basically muscle cell. Sperm cell also can. Okay, but let's say if you say sperm cell, how to explain? Now, the first mark, you state the function of mitochondria first. So why muscle need a lot of mitochondria? Because muscle need energy lor. Sperm cell also say, why sperm cell need mitochondria? Because sperm cell also need more energy lor. Just like I said, why TTC need paper? Because we need to use the paper to print notes. Also, you mentioned the function of the paper lor. So you can say because it is the muscle cell requires more energy. Then I want you to also explain what's the energy for. So this one will be usually another mark if it's a 3 marks question. So muscle use energy for what? To move. So your muscle So muscle punya movement we always say contract and relax. Muscle always contract and relax. So you can say for contraction and relaxation of muscle. If it's sperm, you just say that it requires more energy to swim and penetrate the ovum, swim towards ovum law. Can? Understand? Yeah. Then the next question is the same type of question. Now they ask for component W. Which human cell have a lot of component W, which is our Golgi apparatus? Now, if you go watch my recent YouTube video, I did an exact question like this, like what cell need a lot of what's this fella? Golgi apparatus, right? Okay. Now, before that, remember your ribogo. These 3 organelles, which is your ribosome, rough endoplasmic reticulum, and Golgi apparatus, right? They always work together. Right? So basically, if they ask you what cell got a lot of ribosome, what cell got a lot of rough ER, right? Your answer will be the same. Okay, first, let's go through the function first. Okay, I know some of you know the answer already. Okay, maybe you might ask what is the function of Golgi apparatus? Now remember, Golgi apparatus, we will say will modify protein. Your Golgi apparatus, huh? 。 . 。 。 。 。 。 。 。 。 . 。 。 。 . 。 。 . 。 。 。 。 。 。 。 。 . 。 。 。 。 。 。 。 。 。 。 。 。 。 。 。 。 。 。 。 。 。 . 。 。 。 。 . 。 。 . 。 means they will use protein to make things. Understand? So for example, in your body, there's a lot of things that are made of protein, like enzyme. So your cell, for example, your Golgi apparatus in cells usually will take the protein to make enzyme, make hormones, make blood cells. All these are made of protein. Now, then why does it work together with ribosome and RAF-ER? So these three fellows, they're brother one. They very closely work close to each other one. So ribosome, you remember the function, right? Synthesize protein. Do you remember? Okay, and your rough endoplasmic reticulum, the RO is rough, will transport protein to your Golgi apparatus. So that's how they relate to each other. Ribosome produce the protein. RAF-ER gives the Golgi, so the Golgi can use protein to make things. I like to call like Golgi apparatus like your bakery store like that. And proteins like flour, because your bakery store can use flour to make bread, cake, pastry, and so many more things. So that's what Golgi apparatus do. They use protein to make things in your body. So let's come back to the question here. What human cell need a lot of Golgi apparatus? Basically, human cells that uses protein to make things. Okay? So, for example, just we say enzyme. So where do we find enzymes in your body? Are basically inside your digestive organ, your digestive system, no? Yes, lymphome correction, you correctly, Vernice, very good also. Pancreatic cell. So you can say pancreatic cell, which is your pancreas, is a digestive organ. So this organ need to produce enzyme. So the pancreas punya cell, right, will use the protein to make enzyme to help you digest food. Now, other than pancreatic cell, right, there's a few more digestive organ punya cell that you also can say. Okay, right here also. Or you want to say epithelial cell in stomach or in intestine also can. Epithelial, later we will learn what's this, but epithelial meaning the surface punya cell. Means the skin of the stomach, the surface punya cell lor. Okay, so let's say this is your stomach. This is your stomach. Here's your stomach. Then here's the cell. So the cell inside need to use protein to make enzyme so that your stomach got enzyme to digest food. So in order to make enzyme, I need these three fella. Yeah, people who say goblet cell also can be accepted. But goblet cell, later there will be one question. Okay, then how do we explain? Same, we state the function first. So Golgi apparatus modify protein, right? So it's because pancreatic cell, because this is to modify proteins to secrete enzyme. So what's the protein for? To make enzyme, no? So I modify the protein to secrete enzyme, means to produce enzyme. Okay, yeah. So let's say if they ask you ribosome, your answer will be the same, pancreatic cell or epithelial cell in stomach also can. Then your question is say to modify enzyme, you will say to synthesize protein to secrete enzyme. Instead of modify protein, you write the function of ribosome. Understand? So when you see this kind of question, right, what cell got high density or what organelle, you mentioned the cell already. Then if they ask you to explain, you state the function first. Okay. Next, describe the effect of a cell if the centrioles are not functioning. I'm so sorry, this diagram, the centriole, they draw a bit not like what it should look like. Centrioles, for my students, recently we did the mitosis chapter, we got mentioned about them, right? So centrioles, they are like a pair of cylinder. They are one pair of like myelin and that one. Okay, so which centriole, come, my dear students, centriole punya function is what? Form what? What do they form? They form the line. Remember the strings? What's it called already? Yes, very good. They form the spindle fiber. For those who don't know what spindle fiber, in chapter 6, when we learn cell division, right? When the cell want to divide that time, the cell want to divide that time, they will form these strings between it to help divide the cell. Okay, so if don't have spindle fiber, your cell cannot divide, no? So first they ask, effect of the cell if centriole is not functioning. No centriole, don't have the strings, the spindle fiber. So just say, spindle fiber is not formed. No spindle fiber, the cell very hard to separate. So basically, hence cell division cannot occur. Simple as that. Moving on. State the difference between rough endoplasmic reticulum and smooth. So I think you all should also know, right? Rough. Why is it rough? Because the surface, your face rough because a lot of people, pimple, sorry. Rough ER very rough because a lot of, yes, the ribosome attached to it. So that's the difference. So one got ribosome, one don't have. So how do you write the answer properly is, yes, exactly like that. My friend is very good. So you must say the rough, okay, I just write short form ER lah, ha. But in the exam, remember to spell the whole thing out lah. Has ribosome attached to its surface, but smooth ER does not. Okay, a lot of times you all will stop here, but you must say does not have what also? Does not have ribosome attached to its surface. So you must repeat the whole thing. You all will say, teacher must write the whole thing, meh? Yes, because school is number one is safer. Maybe some teacher they can access, some cannot, but you write definitely is correct. Because got some of my students previously, right, they give me see their exam paper, school exam paper, their teacher give them wrong if they just stop here. Teacher write like question mark, then wrong. So you must continue say if this one got what, this one don't have what. One got hit, one no hit. One got pakai baju, one tak ada pakai baju. So you must say the opposite also lah. If write different function, can okay, this question, they basically want you to state the structure. Is understood one. Rough ER smooth ER, when you ask difference, definitely this one. So this one don't say the function. Usually, okay, if they want to ask this more properly, right, they will say state the difference in structure lah. Usually they will write more precisely. So if you ask structure, yeah, then definitely is this one. But don't write function lah. Ready? Next question. Let's go. Okay. Question 2 is the exact question that I did in one of my recent YouTube videos. But never mind if you didn't watch it. We will do this one more time also. So basically it's Form 5 Chapter 2 plus Chapter 5. Which in Chapter 2, like the organelle cell, how does this chapter relate to Chapter 5? It's because Chapter 5 got one part is talking about the what formation of extracellular enzyme. So for those who attend my class, that this one I actually didn't, I skipped this part. I didn't teach this part. Yeah, so now I'll teach lah. Because when I teach this one, right, it's actually like sort of repeating chapter 2. So I save this for seminar lah. So we get to recap and also do chapter 5 at the same time. So just now we already mentioned how enzymes are formed. You need protein. So if you don't know what's the meaning for extracellular enzyme, extra means outside, cellular means cell. So enzyme that function outside of the cell. So basically all the enzymes, all your digestive enzymes that help you digest food one are actually extracellular enzyme. So just like just now I told you the stomach, right? So your stomach got cells. So when the cells produce the enzyme that time, the enzyme have to come out from your stomach, then only can digest your food. So this is enzyme outside of the cell. Get it? Then, next question, they ask name cell component P and Q, which we already know what are they, right? So this fella, a lot of BG one on top of your nucleus. This one we know already lah, rough endoplasmic reticulum endoplasmic. That's Q, sorry. And P is your Golgi apparatus. Now, in this diagram, you can see very clearly that Golgi and your smooth ER is very different. There's no reason to be confused one. See, smooth ER is right on top of the nucleus, each layer attached to each other, while this one is further away from the nucleus, and each layer are not attached. They're like Wi-Fi shaped like that. Okay, then same like we did just now. Example of human cell that has high density of ribosome. So what kind of cells need protein? The same cells that we mentioned just now, they need to secrete enzyme, no? Pancreatic cell, epithelial cell in stomach, intestine. Next question is our essay question, 6 to 8 marks. Punya essay question. Okay, we're going to aim 8 marks. So describe the relationship between cell component P and Q in the production of extracellular enzyme. So how does rough ER and your Golgi help in producing enzyme? Okay, so let me bring this tree down, this tree further again. Okay, I'm going to mention these three guys again. Now, just now we know already this three further punya function, right? Now, even though this question, right, they just ask about rough ER and Golgi apparatus, but we need to also mention the ribosome if it's 8 marks. Why? Because ribosome is on your rough ER. So even though they just ask about rough ER, but this is actually including your ribosome. So if it's 8 mark essay, we're going to mention the ribosome as well. If essay question can write in bullet form, actually most teacher can accept one. But you don't go write number or bullet in front now. You can write like this, like how I write like that, but you don't write the number in front. Means every sentence you can write one row like that. So you one row, one sentence, then easier for teacher to mark. But you don't need to write the number in front lah. Okay? If your exam until chapter 4, will they ask this? Maybe. Hard to say one. They could ask. So that's why better to know also lah, because got related one. Chapter 2 also we got learn one. Okay. Now, so first you need to know the function of these three organelles. But if you write the function of these three organelles, right, it's only 3 marks only. Right? But now I want 6 to 8 marks. So I go to write more points elaborating each of the organelle. SPM format allows us to write in point form. Okay, like I say, you actually can write like how I write here, but you don't write the point and bullet lah. Means you can write sentence by sentence. And your sentence don't have to be complete. I mean, because this is not English, it's bio ma. So you don't have to say, the relationship between cell component and P is, okay, you don't have to write that. You can straight start your answer with because, you can just straight away start your sentence. Cannot or she wants essay, then you follow your teacher. If your teacher mark your paper, you follow what your teacher write lah. The teacher say lah, because your teacher mark your paper ma. Okay, but usually you can write one sentence, one row. Like for example, okay, later I'll tell you more lah. Okay, let's start first. Okay, now number 1, okay, we start with the ribosome on your rough ER first, because protein is the first thing you need ma. So point number 1, I first mentioned, ribosome on cell component Q, which is your rough ER, synthesize protein. Okay, come on guys, we write this together. Okay, then once you got protein already, we know the rough ER will transport the protein, right? But before that, before you can transport the protein, okay, for those who got watched my video one, can you tell me what you need to do first or not? You can see here the diagram, a lot of these. What are these actually? So before you transport protein, we need to take the protein and package into this thingy called the, called the transport vesicle. If you go watch my video, you know what's this. So transport vesicle is like lorry like that. they will transport the protein here. That's how the rough ER transport the protein. Okay, the words that I use here is a bit different in our textbook lah, but the keywords are still there. Means I make it slightly more simpler a bit, so they're easier for us to write and remember lah. So we say in Q, proteins are packaged into what we call as transport vesicle. Then this transport vesicle barulah transport the protein to your Golgi apparatus. Okay, you're right, Right. And then next, and it will fuse with the Golgi apparatus. So now transport vesicle take the protein to the Golgi already, right? It will fuse with it. Okay, now when you write in actual exam that time, you don't go and separate the sentence out lah. So when you fuse with it, you just write behind here and fuse with it lor. In classes, I will always write like point at that. It's because I want to show you the points. Yeah, but when the points is in one sentence, you just write it one sentence, means you don't go and separate the sentence lor. But you can write one sentence, one row lah. Easier a bit. Easier for the teacher to mark also. But if like this one, you don't go and separate, you just continue behind here. Okay? These are a few tips that I can tell you. Now, so now we're done with these two further already. I got the protein already. I transport the protein already. Now I reach the Golgi apparatus. So what does Golgi do? Take the protein, modify to enzyme. Means I use protein to make things. So that's what we mentioned. P, which is your Golgi apparatus, will now modify the protein into enzyme. Once you modify the protein into enzyme already, you got to package it one more time. Why you want to package one more time? Okay, is I make bread already. I take a flour, I make bread already. Before I deliver to my customer, also I need to package it. Okay, this time I will package it into another vesicle. Can you guys tell me or not? What are these another vesicle? Because now I want to transport the protein out. So this another vesicle that I will package is called the wow, a lot of people know they very smart yeah, secretory vesicle, very good. So what does this secretory vesicle do? It will take the enzyme and then they will fuse with the plasma membrane here and then release the enzyme out. So that's how I get extracellular enzyme. Okay, so secretory vesicle is the next thing. Also, after I got this enzyme ready, I package one more time. This time I package it into secretory vesicle. And then, okay, you write this one first, secretory vesicle. Okay, then this secretory vesicle, like I said, will stick to the membrane here. So we will say it will then fuse with the plasma membrane. So secretory vesicle then fuse with the plasma membrane. After I fuse already, then only I let the enzyme out. So we will say and secrete the extracellular enzyme out of cell. So this is 8 marks in total. 8 marks actually can donate to write about the DNA, RNA. Okay, if it's 10 marks, then only you need to mention the DNA, RNA, which you must say the DNA, transcribe the transcript into RNA, RNA then translate, undergo translation at the ribosome. But usually they won't ask that one, that one not important one. Can understand this one. Any questions for this? Can, if can, we move on. Okay, so next, okay, I skip to here. Okay, in my video also I got do this one, so I just mention it real quick. So now they ask the opposite. Okay, you can go, if you got these notes, you can look at page 4, page 21, sorry. What will happen to production of extracellular enzyme if P and Q are absent? So same, if I tada rough ER, tada Golgi apparatus, what will happen? So out of these 4 marks, right, one of the marks very easy one, which is, do you think you can produce enzyme or not? Answer, of course, is no lah. So if you mention extracellular enzyme cannot be secreted or produced, then you get one more idea. Okay, but you got to explain why I cannot. Same. You just mentioned the P and Q punya function cannot occur. For example, no Q means no ribosome, no protein. No protein means you cannot transport it here. So the blue color point that I write here is about your rough ER. So no Q, no ribosome to synthesize protein. Okay, you can copy this down. Then no protein, then you cannot transport it. So proteins are not transported to P. And then if you don't have Golgi, even though you got protein, you cannot modify it to become enzyme. Right? So we say that proteins cannot be modified into enzyme. Hence, enzyme not secreted. So very simple one. You just mentioned the function cannot occur. No ribosome, no protein. No rough ER, cannot transport. No Golgi, cannot modify. So therefore, no enzyme. Boleh Can Okay. If can, then look at page 19, question 3. Okay, I'll give you a few seconds to copy. Okay, we take a short break because I want to take a toilet break. So we break around 3 minutes, then we continue, Ken. All right, let's look at this question. So you can see here I will do a lot of those kind of exam, kind of trial questions. So this is also an actual trial question they ask. Diagram 1 shows 2 cell components that are abundant in a plant cell. Okay, so we already know this is mitochondria, quite obvious. What about Q? Q is oval shape also, but you can see inside is completely different, right? So this one mitochondria inside is like curly like that one. This one, a lot of like one BG things stacking up one. That one is your page. This is page number 19, question 3. Yeah, this is your chloroplast. Now here they didn't ask us to write the cell punya, the cell component punya name. They want us to name the cell which has dense with components P. Dense means got a lot of P and Q. So now this is plant. So you cannot write muscle cell, you cannot write sperm cell already, because this is plant. The question here got state very clearly already, plant. So what plant cell need a lot of mitochondria? Can you remember this? This one in class, I didn't really mention that clearly one, but I got teach before one, if you remember. The leaf, no, not leaves. Okay, the answer, a lot of people know, yes, correct. Mary's palisade is chloroplast. So Mary's stem cell or Mary's spermatic cell. Yes. For those who don't know what is that, okay, in a plant, you have the shoot tip and the root tip. Okay, Mary's spermatic cells are located here. These cells, what do they do is, right, they will keep on cell division. Divide. And that's how they can grow longer. Same also on top here. So cell division, you need actually a lot of energy. Like that. Like that. So since here you need to carry out a lot of cell division, the cells here also need energy. So your answer is Mary stem cells or Mary spermatic cells. You write ATIC behind, also correct one. Mary stem cells or Mary spermatic cell, same. Both also can. Okay. And what plant cell need a lot of chloroplast? The leaf inside got one cell that carry out photosynthesis one. You must say palisade, mesophyll cell. Last time we also got learn this before in science one. Yes, palisade. Okay. Why component P is dense in the cell name in 1A? So why meristem need a lot of mitochondria? Okay, so I already taught you how to answer this kind of question, right? Number one, you state the function first. So just like muscle cell, just like muscle cell and sperm cell, we need energy ma. So because it requires more energy. The energy buat apa? Just now already say lor, cell division ma. So to carry out cell division or mitosis also can. Can. Next, one similarity and one difference between P and Q. Now, this one, they were more commonly to ask similarities. Mitochondria and chloroplast got what similarity? Anyone remember or not this one? Wow, gang, all these. Wow, Lim Hong also know. Yes, the membrane got two-layer membrane. Surprisingly, you still remember very good. Yeah, so you can see, right, mitochondria got one layer over here and then second layer means got two layer one. Chloroplast also see one layer, two layer. So they have what we call as double membrane. Of course, you must say both. Both have double membrane. Differences. Okay, the difference is basically the function. This one, the difference is mainly talking about the function for this chloroplast and mitochondria. So organelle P, which is your mitochondria, right? Yeah. So these two organelles, right, they carry out opposite punya process. In living things, right, these two processes actually terbalik one. Which now we know mitochondria generate energy, but what's the process of generating energy? Sorry, cannot read your name. What Candy? Okay, wherever you are. Okay, correct. Olives also yes, very good. So the oxygen plus glucose equals energy. That process. So that is how energy is produced in living things. So that is basically cellular respiration. So carries out. Okay, cellular respiration. Sorry, got some weirdo interrupt us just now. Okay. And then organelle P, we will say carries out photosynthesis. Can I write the chlorophyll thing? This one better don't. Yeah, the difference memang they mainly they want you to actually state the process that they carry out. Yeah. So don't say the chlorophyll one. The one I'm not too sure whether they can give you correct or not, but better don't lah. Okay. So that's the difference and similarities. All right, let's go. Next one. Okay, this question a bit not really related to what we learned in chapter 2, but it's actually like more like a Form 5 photosynthesis punya question. Okay, now why we just do 2 marks only. So they say there is activity of quarry area which cause air pollution. Quarry means the lombong mining lombong biji lima. So they ask, explain the effect of quarry activities. Okay, let me remove you also. Explain activity of quarry activity towards function of component Q. Anyways, yeah, this is the problem if we have no public class open to all public, right? So weird people also will sometimes come and join us. Anyways, where was I? Oh my god, so annoying. Okay. So now here they say got air pollution. So mining, right? Mining means a lot of Oh yeah, lor, they all keep coming with weird names, oh my god. They got nothing better to do, very lifeless. Okay, next one come on. I want to scold him. Okay, anyways, air pollution means there's a lot of how to say dust in the air. Because mining, you mine the rocks explode a lot of dust. So if there's a lot of dust in the air, they ask, I appreciate dust. How does it affect the component Q, which is chloroplast, to carry out photosynthesis? So we know chloroplast carry out photosynthesis, right? So with a lot of dust in the air, how does it affect a plant carrying out photosynthesis? It blocked the leaf, yeah. So mainly there is no sunlight. Exactly. So that's what you can say. So how does it affect the function of component Q? So you can say the quarry activities or the air pollution reduces, you can say, light intensity lah, because if there's a lot of dust in the air, the sunlight cannot pass through. Like when it's haze that time, or your surroundings are all like very cloudy like that. Okay, because they block the sunlight law. So therefore, how does it affect the function of component Q? So you can say law, the rates of photosynthesis in component Q decreases. Yeah, but this is more like a Form 5 question, but never mind. Okay, then let's jump to page 24. Question 6. Once you're done. Okay, this one, I just do C can already lah, because after that, all the questions are quite repetitive already. So there's no point doing the same thing over again. You just practice yourself. Now, explain one characteristic of X. Characteristics of X. X, this thing you can see, right? Quite obvious, is our mitochondria. Now, if they ask characteristics of mitochondria, remember or not? What is the characteristic of mitochondria? Double membrane, okay, double membrane cannot say already for this one because double membrane got nothing to explain. If they just say state the characteristics, okay, yeah, you can say double membrane, but double membrane got nothing to explain. Double my double lor. Okay. So another thing, the first layer membrane is like that. Yes, but the inner one is folded, remember? Now this one got function to explain. So you must say has folded or inner folded membrane or folded inner membrane. Yeah, either way, correct also. And in bio, I already mentioned to you, whenever you see got folded surface, right? Compared to a flat surface, folded surface, yes, you also know, right? Got more surface area. So you can say, which increases the total surface area. Okay, but don't just stop at surface area. Explain what's the surface area for. If it's 3 marks, you need to mention. So basically, more area, you got more place to increase rate of cellular respiration. More surface area, more respiration. And also you can say to produce more energy also can. You want to say after that to produce more energy? Yeah, also can. Done. So yeah, remember, mitochondria, they quite like to ask this one, the folded surface, that is their characteristics. All right, then we move on. Okay, I will skip here and there a bit. Okay, we go to page 26, C. Okay, this one I do C can already. C, now, the cell has been infected with a bacteria. Explain the cell component that can destroy the bacteria. So let's say I got a cell got bacteria come and attack it. So what cell component can help destroy this bacteria? White blood cell. Okay, white blood cell, you're not answering the question because the question says, okay, even though this is white blood cell, yeah, white blood cell help destroy bacteria, but what component inside the white blood cell can destroy this bacteria? So they want cell component, right? They didn't ask the name of the cell. Yep, inform correct. Yes, of this very good. So this organelle start with letter L. So just now we got mentioned one, remember got one organelle that is like this wrong one. So that fella is called the lysosome. No, not food vacuum. You must say lysosome. Lysosome is basically this thingy. Now, they will secrete a type of enzyme called lysozyme. So this enzyme punya name sounds the same like lysosome. Yeah, so must say lysosome. Can Now, when I say this fella produce enzyme, right, then somebody will say, teacher, just now we also learned Golgi apparatus modify protein to enzyme. Can we say Golgi apparatus or not? Okay, the answer is no. Because Golgi apparatus, yeah, I use protein to produce the enzyme, but the enzyme is mainly to give transport outside of the cell for your body to use. So lysozyme is the one that will actually go and kill the bacteria using its own enzyme. So you must say lysosome secrete lysozyme. Okay. Then they ask you how to destroy the bacteria. You can say which destroys and kill the bacteria. If say hydrolytic enzyme, is it correct? Can, but I would prefer you to write lysozyme lah. Like actually, hydrolytic enzyme actually most of the time they accept one. Yeah, but I think lysozyme would be a better answer. Okay. If say hydrolyze can, which hydrolyze the bacteria, yeah, can also. Actually better, right? You can say, yeah, which hydrolyze and kill the bacteria. Hydrolyze means you break down the bacteria lor. Yeah, actually don't say destroy that because you repeat the question when you say destroy, so we say hydrolyze or break down the bacteria lor. Thank you. Yeah, would be a better answer actually. All right, then we do one more cell component punya question. Okay, we do a lot already. So this is the last cell component question. Then we need to move on to the next topic already. All right, this one is a plant cell. Now, the one that I want to focus is question B. Okay, wait. Which one? How to spell that? I'm so sorry, my writing a bit ugly here. Okay, write properly again. Hydrolyze. Hydrolyze means breakdown. Yeah, I can actually do it here. I done already. Oh. Hydrolyze means breakdown, destroy. Yeah, breakdown. Hydrolyze means breakdown. Okay. All right, let's continue here. Should be very peaceful now already. Now, anyways, give two difference between cell component Q and R. Cell component Q and R, cell component Q and R. Okay, so Q and R. Q is actually pointing at the plasma membrane. And this is the cell wall. So you all know, right? Plant cell got cell wall, then got the membrane, but the membrane, we will write it as plasma membrane, no longer cell membrane. What difference? Now, they got two difference you can write. One is the structure. Number two is their characteristics. Okay, we talk about the structure first lah. Now, what are they made up of? Cell wall, we learn in chapter 4.1. I also recently got post one chapter 4 video, recently got mentioned one. Cell wall made up of what? Cell wall is made up of cellulose, right? Yeah, your fiber, the polysaccharide. Remember carbohydrate got mono-di-poly, yeah, so it's the polysaccharide. Now you remember. So yes, so that is what R is made of cellulose. Okay, what about plasma membrane? This one in chapter 3 you got learn one. Plasma membrane are made up of two things. Fully and semi-polar, that's the characteristics. Okay, that one's the next one. Cell membrane and plasma membrane, is it different? Cell membrane and plasma membrane is the same, but in Form 4, we use the more canggih punya word. We use plasma membrane. We cannot write cell membrane anymore. It's the same thing, but we cannot say cell membrane lah, huh? Yes, it's made up of two things, the protein and phospholipid. Very good. Yes, you remember your channel carrier protein, they got the phospholipid. Exactly. So P is made up of protein and is it P? No, it's Q. What is P? Q is made up of protein and phospholipid. Two, not two. Haven't read the other one. R is made up of cellulose. Number two, the characteristics. Okay, cell wall is fully permeable. Everything can pass through it. But like we learned in chapter 3, plasma membrane only allows certain substance to pass through. Remember, only allow what? Small and uncharged. If you like water, if you are large, like glucose, you cannot enter. So yeah, that is the second characteristic. So Q is a cell wall, right? So right, Q is plasma membrane. Yeah. So Q is, we will say, selectively permeable, while R is fully permeable. So let's say here's the cell wall. Secondly, here's the plasma membrane. So everything actually can pass through it, the cell wall. Fully permeable. But after you pass through cell wall, right, only certain things can pass through the membrane and enter the cell. Strong and rigid. Okay, cell wall is strong and rigid, but cannot lah. That one cannot. Better don't. Okay. Right. Then let's finish off this one. After we done this, then we done with organelle cell structure already. Now 1.2 shows organelle Q. Organelle Q is basically your rough ER ribosome. Okay. Now, and then why cell goblets in the respiratory tract contains high density of Q? Now, again, we are talking about the three brothers, ribosome, rough ER, Golgi. So other than your digestive system punya cell, the pancreatic and intestine cells that we mentioned just now, now there's another cell called goblet cell. Goblet cell produce what we call as mucus. Just now earlier, a lot of you also give me this answer. Ken, correct also. So mucus, okay, if you don't know where is respiratory tract is basically your trachea, your airway, the air that goes, the airway that, transport air into your lungs. So inside your respiratory tract, right, there's lots of this cell called goblet cell. Goblet cell actually produce mucus. Okay, your nose, the mucus. So this mucus is actually made up of protein, just like enzyme. So same. We will say, state the function first. So I want the ribosome is because I want to synthesize protein. What's the protein for? Just now it's to make enzyme, right? Now it's for mucus, to secrete mucus. Hey, this one can write energy. This one is not mitochondria. No energy. S has pigment found on the thylakoid membrane. Okay, thylakoid, we learned about this in Form 5, but thylakoid basically is the structure inside your chloroplast. Then S is your chloroplast. Okay, now we don't care about this thing called thylakoid. We'll learn it next time. So pigment inside your chloroplast, there's this green pigment. Organelle Q. Q is actually pointing at the ribosome stroke rough ER. But rough ER is transport protein. So transport protein not really like suitable for the explanation here. So we just say it helps to synthesize protein. So the protein can be used to make mucus. Okay. Now, back to this question. The pigment means like color. Chinese 是颜色素啊. So what is the color pigment inside your chloroplast? So it's actually this green color thingy called the chlorophyll. Yes. So if you're wondering what's the difference between chloroplast and chlorophyll, chloroplast is the entire organelle. It's called chloroplast. The green color substance inside is called chlorophyll. So we name the pigment, which is called chlorophyll. The function of chlorophyll is to absorb light photosynthesis. So it absorbs sunlight to carry out photosynthesis. Moving on. Okay, we're done with organelles already after that. So the remaining one, you all can just try to do yourself. Answers at the first page got this QR one. You can just scan it. Okay. Right. Then let's go. 29. Not done. Sorry. Okay, let's go. Let's go. Next. Now, this is the cell organization, tissue organization question, which you all remember what is that, right? The cell form tissue, tissue form organ, organ then form system, system then finally form an organism, that one. So those are what we call as cell organization in human or tissue organization. So whenever you see the word cell organization, tissue organization is basically this stuff. Now, we will focus more on tissues. Question meaning like to ask tissue more, which is harder a bit because organ you all know, right? Brain, stomach, intestine. But tissue maybe slightly harder a bit. Okay. There are two main tissues that they commonly like to ask for this cell organization, which is epithelial tissue, means your surface tissue, skin tissue, and muscle tissue. Okay, so this tissue, can you guys tell me what it is? Can recognize? So in your intestine, got what tissue? Got villi. Villi is formed by this tissue. So this one you must say epithelial. It's actually your epithelial. Now your small epithelial means in Chinese pi means skin, surface. So let's say this is your small intestine. There's a lot of these cells on the surface of your small intestine. So these cells, which is the epithelial, forms the so-called villi that we did last time. Okay. Now the epithelial tissue on your small intestine, right, is a bit unique. It looks something like this one. The surface is like that one. So there's a lot of it like that one now. Okay. So this tiny projections, right, is called the microvilli. So the microvilli of this epithelial is to help increase surface area to absorb nutrients. Later got one question asked about this one. So, and also other than absorb nutrient, right, the epithelial tissue in your small intestine, right, just like we mentioned earlier, help to also secrete enzyme. Remember, epithelial cell in stomach and intestine. Yeah, so this cell also can produce enzyme to digest food. They also can absorb nutrients. Okay. Now, so I already basically have you done this cell, tissue, organ system. Then they ask, state the name of structure V. So what system is, or I mean, what organ is this? This organ is a... organ is that? Intestine, right? More specifically, small intestine. Moving on. Instead of cell P, state one example of another cell that is found in structure V to allow the peristaltic action to occur. Okay, so other than epithelial, they're asking, what's another cell? Okay, remember, tissues, they are made up of the same cell. So if this is epithelial tissue, this is epithelial cell. So they're asking, other than epithelial, what's the other tissue or cell that can be found in small intestine that help in peristaltic action? You all still remember what's peristaltic action, right? Peristalsis, when you swallow food, that time, this group movement, they push the food one. Still remember, right? Okay, so what cell, what tissue helps in peristalsis, peristaltic movement? So how many? So okay, you think about it. In order for your intestine to move, in human, right, movement use what tissue? Yes, Jaden, very good. We use muscle. Yes, exactly. Kai, Olive, very good. So, however, later we will do one question. There are actually three types of muscle in human. The muscle inside your digestive organ, your internal organs, right, is mainly smooth muscle. So I want you to write smooth muscle, not just muscle. Our next question will be doing muscles, okay? Smooth muscle cell, since they are cell. So one mark, settle. But if let's say they ask you to explain it, every muscle in your body got one role: contract, relax. So it contracts and relaxes to produce peristaltic action. Okay. And moving on, they ask, name the system shown in level 4, system shown in level 4. So obviously, different tissues form an organ. Then you have different organ forming a system. Intestine obviously is your digestive system lah. I mean, come on, this is too easy. Okay, this one I don't want to do lah, because this is actually Form 4 Chapter 9 is about digestion one, the fiber one. Yeah, so this one I will skip. We quite limited time today, so yeah, this one I don't want to do since it's not really related. Next, they say this one they were doing just now. The inner wall of small intestine is layered by a lot of tiny projections adapted to carry its function. Explain the advantage of this adaptation. So the tiny projections that I was talking about are this. So these tissues on the surface, a lot of these bulu thingy one called the microvilli. So they are asking here, what is the advantage? Okay, so the question didn't ask me to name this thing. So don't name it, there's no marks. So what does it do? So if your surface is like this compared to like this, got what advantage? So just like the mitochondria, it provides larger total surface area. Yes, or increase the total surface area also can. And what is it for? More surface to absorb nutrients, so you allow nutrients to come in easily to absorb nutrients efficiently. Then, next page. This is muscle tissue punya question. So we done epithelial just now. Okay, now we do one muscle one. Okay, other than the epithelial tissue here, right? Another epithelial they could ask is your epithelial of your skin. Okay, your skin punya epithelial is a bit different than the epithelial you just saw just now. Your skin punya epithelial, right, actually is like this one, a lot of layers one. So if they ask you the characteristics, right, just now that one is a lot of microvilli absorb nutrients, right? For this one, you must say it is thick to prevent entry of microorganisms. So your skin punya epithelial slightly different lah. Okay. Then next one, page 31. Name tissue L, M, N. So these three are muscle tissues. Okay, let's name them one by one. Now, the first one, it looks like sticks, like bones. So these are muscles that are attached to your bones, the most common muscle in your body. So that muscle is called skeletal muscle. So they literally look like bones. Yes. And this one, now sometimes this one, right, they might draw a bit quite similar to this. So how to see carefully is, right, they also might draw like this, like sticks, bones like that. But this one, they got like branches. So that means they will branch out like that one. You have branches branching out like that one. Okay, sorry, my drawing a bit ugly. But yeah, you can see they like branch out one, like branches. So this one look like ginger. Okay, yeah, you can remember like that also. Up to you. So for me, look like branches. So this one is called cardiac muscle. Cardiac, you know what the meaning of cardiac, right? Cardiac means it's found in your heart. This is your heart punya muscle. Yes, you're correct. So this thing is your heart punya muscle. Skeleton is your bone punya muscle. And the last one is what we mentioned just now, the smooth muscle. Smooth muscle are basically found in all your internal organs except for heart. So especially your digestive tract, your esophagus, your stomach, your intestine. This is all smooth muscle. Your blood vessel also is smooth muscle. Heart is cardiac law. Okay, so let's write the function here. Now remember, every muscle contracts and relax. So I don't write contract and relax already. So for example, skeletal muscle is to contract and relax to produce movement. So we rely on skeletal muscle to move. Cardiac muscle basically say not contract and relax for heart to pump blood. Smooth muscle we did just now already, contract and relax to produce peristaltic movement. Pump, what is this? Pump blood. Next, how tissue L is formed. Now, cell forms tissue. Tissue form organ. They like to ask you to explain tissue and organ a lot. Okay, so which definition, no? So this question, right, is actually use the definition to explain. So they won't ask you too direct and ask you, what's the definition of tissue L? Now they will ask like that, how tissue L is formed. Now, 2 marks. Number one, we first start by saying, what is it made up of? Okay, cell differentiation, no, not correct. It's actually cell specialization. Okay, but other than cell specialization, first, I want you to write like this. Tissues are formed by a group of cell. A group of similar cell. Remember, epithelial cell form epithelial tissue. Smooth muscle cell form smooth muscle tissue. So you can say it is made up of a group of similar cell. Okay. And then which work together to perform a specific function. Okay, this one don't need to write the cell specialization. If they ask you like that, right, explain how a tissue is formed, use the definition to explain. Okay? Yeah. But just now the cell actually is specialization and not differentiation. It's a process of cell forming tissue. So sometimes they like to write here like that. What process is this? This process is called cell specialization. Okay? Done. And let's say if they ask you organ. Now, organ also made of tissue. So if they ask you definition of organ, how organ is formed, or why stomach is an organ, okay, it's also write the definition. So, but organ now is not made up of same tissue. Organ is made up of different tissues. So, like just now here, our small intestine, right, got epithelial, also got muscle. So if they ask you organ, we say made up of different, right here, they are made up of different tissues. Then the second point is actually the same, work together to carry out specific function. Like they usually will ask tissue and organ. In exam, will they ask definition for differentiation and specialization? No, they won't ask that. They won't ask one. They want too difficult already lah. So it's just, you need to know cell form tissue is specialization. Differentiation is another thing. Differentiation is what we recently learned in chapter 6.1, whereby the embryo cell, you know, when you're embryo, then you have a group of useless cell. After when you become baby that time, this embryo cell will then differentiate, means become a specific cell. Like you'll become muscle, become bone, become nerve. Yeah, so that is called cell differentiation. It's a different thing, totally different. And this question, we already done it almost, a similar question just now we done already. How tissue N helps small intestine to carry out its function. So tissue N is basically this, your smooth muscle. Oh, I forgot to tell you how to identify smooth muscle just now. So smooth muscle, number one, it looks like eyeball shape. So it's very different one. So easy to recognize. Number two, right, the surface or don't have like line that one. That's why it's smooth for. This one you can see a lot of lines like that. That's why it's rougher. This one don't have. So that's why it's called smooth. So they're asking you how does this helps, how tissue N helps small intestine to carry out its function. So you can see this is a smooth muscle here. So just like every muscle, it contracts and relaxes. Then like we've been writing for a few times already, to help push like that. So it's called peristaltic action to produce peristaltic movement. And then what's peristaltic movement for is to push the foot, to help push food along the small intestine. Can I say involuntary action instead of peristaltic? Cannot, because this question specifically want you to answer how does it help the small intestine. So the specific involuntary action here is peristalsis. So you have to say peristaltic action or peristaltic movement. If you write involuntary, definitely is wrong for this question. You have to be very specific lah, because if they show you small intestine, push the chime, just say push food can already lah. Okay, then this one you got another one, you try yourself lah, so you can see, done already this one. There's epithelial, then again got smooth muscle, look at eyeball like that, so it's in your stomach. So same more, stomach also got epithelial to produce enzyme, but the epithelial in stomach won't absorb nutrients. Got smooth muscle, same, peristaltic action, same thing. Okay, then page 12, we just go through a little bit this diagram here. Can you guys recognize what is this cell J and tissue K or not? So of course, tissue is made of similar cell, so these two is the same thing. So what's this showing you here? What's it showing us here? Yes, all this correct. It's actually, yes, Jaden, very good. This is your cardiac muscle, yes. So you have cardiac muscle tissue, cardiac muscle cell. Remember, tissues made of similar cell. You can see got branches one. So if this is cardiac muscle, what's the organ it will form? Yes, heart. Heart belongs to what system? Is your correct blood circulatory system, and then form an organ is a human. Okay. All right. Our final half an hour, we will do the last topic, paramecium and amoeba. So the plants one, I will skip lah. I won't do plant punya one today. We will then skip to our amoeba paramecium. Let's go. Okay, let's look at page 39. Last few already. Come on, guys. We're almost there. Right, this is also a recent trial paper. You can see pair up 2024. Amoeba. Okay, amoeba, right? Labeling first. Now, there are three round things they will commonly ask you to label, which they don't look exactly the same, of course, so you must know how to recognize. What organ system is the most asked in exam? Digestive one, not the one that we just did. You know, your stomach, your intestines. They quite like ask these two a lot. Yeah, so you pay attention on these two. Just now, the heart one also they can ask one lah. But digestive organ is the one they ask the most. So we did the digestive just now lah. Moving on. Yeah, we have amoeba here again. So this round black one obviously is the nucleus. What about the round and white color one? X. What is that? Water vacuum. Okay. So you got one part of it correct. So yes, there's water inside. Very good. But the full name is called contractile vacuum. Okay. Now, why is another vacuum? This vacuum got things inside one. You can see this one is water, that's why it's like white color, but this one got something inside one. So it's called, yes, foot vacuum. And Z, their arms. So this is the most unique feature of the amoeba. That's how to differentiate. Yeah, I got teach you all before. Foot means got thing inside one. Empty one is water, is contractile vacuum. So if you look at page 38, right, this one is like a more accurate diagram of an amoeba under a microscope. You can see, white color one, this is the contractile vacuum. And the black color one, nucleus. Then got things inside one, that's the foot vacuum. Yeah, this one you try yourself now, Okay, so Z is their so-called fake arms called the pseudopodium. So if you understand a bit of Chinese, so means arms. It's like this, their arms like that. Next question, they ask us to draw the amoeba, which they are showing us the feeding process, the makan punya process called phagocytosis. So if they ask you what's this process, you must know how to say it's called phagocytosis. So you can see they use their pseudopodium, makan the food. Once the food is inside them already, right? the food vacuole will form around it. So the next picture you're going to draw here is an amoeba, whereby the food is trapped inside the food vacuole. Label, like the question says, this is your food vacuole. And then lysosome will be something that be attached to the food vacuole. So you can draw one round thing like that. Because lysosome, just like just now, we say produce the what lysozyme to go and digest the food. Remember? So yeah, to digest and kill the bacteria. That's what they're doing here. They're using enzyme to kill it, the lysozyme. And then we just follow the question. I've got one nucleus over here, right? Yeah, so we just draw the nucleus here. All right, I want you to find a blank piece of paper or just find an empty space. We're going to practice write the phagocytosis process one time lah. Your first term, they quite like to ask this actually. Okay, so the process, you can look at the diagram here, okay, which is the same. Let's practice our phagocytosis, around 5 marks punya explanation. So number one, if got food, they will use their soda podium like that. So how do we explain this? We will say the amoeba extend its pseudopodia. So you can say pseudopodia. You want to write pseudopodium also can. It's basically singular plural. One pseudopodium, two pseudopodia. And engulf the food. I just write the food particle lah. Engulf means like wrap around it. Mengepung in Malay we say wrap around it. And then number two, like what we draw just now, once the food is inside the amoeba, food vacuole will form around it. So we say in the amoeba. So every time you write amoeba, you must write the SP dot. It stands for species. That's how you write their scientific name. Food vacuole forms around the food particle. My mic, hello. Got sound? 1, 2, 3, testing. Can all right, my mic is moving there. Hello. Okay. I just now no sound. My mic also sleepy already. Very robot. Plural and singular won't have any effect. You can just write anyone. This is not English. They won't judge your grammar one. And then the lysosome will attach to it. So we say lysosome attaches or fuses with food vacuole. And just like we did earlier, lysosome inside got a type of enzyme called lysozyme and secretes lysozyme into it. So you mentioned lysosome, 1 mark, lysozyme, another mark. What does lysozyme do? Digest the food, hydrolyze the food also can, which digests the food particle. Settle. Let you rejoin. Okay, you can rejoin. Wait for you. You want to screenshot the answer first? Hello. Do you have to write elimination of the waste product? Okay, usually when they ask you to explain phagocytosis, right? It's just like that because phagocytosis means eat ma. So when I explain this whole thing, right, I actually done explaining the eating process already. So the elimination process actually is not part of the phagocytosis, but very commonly you see that textbook, a reference book got mentioned the eliminating part, like you can see in our diagram here, right? Yeah. This one usually not very important, okay, but the elimination part actually very easy. You just have to say, okay, the last one, you just say after that, amoeba just moves. Yeah, sorry, I paused the screen already. Moves away, leaving undigested food behind. That's it. But usually phagocytosis is like that. Like the main process is 1 to 5 lah. All right. Approaching the end already. Next, continue. Everybody done with this? Question C. 3, 2, 1. How amoeba adapts if the environmental conditions are not favorable? Now, paramecium amoeba, right? They behave differently when the condition is favorable and not favorable, means they reproduce differently lah. Now, sometimes question won't be so direct and say, oh, not favorable. Sometimes it gives example of the surrounding, and you got to know whether is it favorable or not. So what do they don't like is basically not favorable condition is basically they don't like high temperature, hot water, for example. They don't like bright light, means like sunlight. They also don't like acid, which is low pH. And they also don't like dry places because remember, they live in water, freshwater pond, as the question here mentioned. So those are not favorable conditions. So if it's not favorable, how do they reproduce? There are two ways they reproduce. One way is not favorable. Yeah, they form spores. So you can say they reproduce asexually. But you write asexual, you got no mark one. I just write more complete. But you must say through forming spores. Forming spores is the main one that got marks law. Okay, can hear me already? I'll leave. Okay, then what if it is favorable? Favorable, how they reproduce. So just like what we recently learned in Form 4 Chapter 6, mitosis, just one split into two like that, right? Yeah, okay, we cannot say mitosis, must say binary fission. But binary fission basically uses the concept of mitosis, but the method, you must say binary fission. All right, settle. Let's go. Another one, next page. Very happy because it's going to end class already. I can enjoy my weekend. Hey, do you know, guys, actually, we teachers also look very forward to end class one as much as you all. You go to end class, you're very happy one. Alright, same one, okay? Okay, come. Page 40, continue. Sorry, I'm being too honest. Now, structure P, okay, the white color thing, contractile vacuole, you know what's this, Can write yourself. Okay, we explain the function over here. How structure P helps the organism. Teacher question won't ask conjugation punya explanation. No, they won't ask conjugation punya explanation one. Too difficult already. But later, yeah, later got one question we need to write conjugation. Okay, now how structure P helps the organism to overcome problem of the cell from being burst. Now, number one, they live in fresh water. So fresh water means ada banyak air di luar. In chapter 3, condition that have high water potential, remember called what already? High what? Yeah, hypotonic. So means water will keep on going into the cell because a lot of water, so water always go from high to low water potential. So if water keep coming in, right, the amoeba will like keep expanding, it will burst. So they ask how P can help it to overcome. Yeah, so someone mentioned osmoregulation. Now, osmoregulation is what the function of contractile vacuum. So they carry out something called osmoregulation. But for this question, right, you write osmoregulation, only not enough marks lah. But osmoregulation is not the main thing here. Okay, but it's one of the points you must say. Or actually, we can say like you can say by carry out osmoregulation. So the meaning of osmo means water. Regulate means you control. Basically, it's like your bladder like that. When you drink too much water, your bladder will accumulate all your excess water and then you go toilet. you release out. So this is called osmoregulation. We control the water. So now we're going to explain how they do that. Okay, it contracts from time to time when the contract regular expands. Okay, yeah, something like that, Alicia. Okay, but first, one by one, first we mention the excess water enters it first. Okay, so excess water, just if you drink a lot of water, the water enter your bladder. So excess water diffuse. Okay, where's the excess water from? You can say in cytoplasm. First diffuse into structure P lah, which is your contractile vacuole. Okay, I just write CV contractile vacuole. So then remember you must write this. When it enlarged until maximum size, baru it one shot contract. So it's just like your bladder. When you got water inside, right, you don't keep on going to toilet one. You only will go to toilet when the bladder is full. Then only you got the urge, then only you go and pee and release the water. Right? So you must write this point, remember, when contractile vacuole enlarges to maximum size, then contract. Yeah, you write osmoregulation got 1 mark one. But for this question, you don't need to write that one because got more than enough points already. It will contract and expel excess water to surrounding. Ta-ta! And then question C is the opposite. They also like to ask this. What will happen to structure P if the organism is put into a highly concentrated salt solution? Now, just now, freshwater banyak air is called hypotonic. So now, yes, very good melody. Outside now is the opposite. Now it's very salty, a lot of salt. Means there's lesser water, so it's called hypertonic. So meaning now, water is going to come out. So what will happen to this fella? Okay, number one, yeah, it will shrink, but since it's 3 marks, now how are we going to explain this? First, we can say this contractile vacuole will contract slower already. Because there's a lot of water, they keep on contracting, expel the water, expel the water, expel the water. Now there's not much water, so it will contract much more slower. And then it will come to a point whereby there's no more water entering it. Because all the water come out already. Then only you say it will shrink to minimum size. Just not expand to maximum size, right? Now become minimum size. Cannot say die, because they're asking about P. They didn't ask about the amoeba. So number one, you can say water diffuses out of 3 marks, actually this one, out of, as we explain, your amoeba by osmosis. Okay, sorry, I write here, I still leave not enough lines. Then you can say, therefore, the rate of contraction of your contractile vacuole, I write here CV contractile vacuole, reduces. So you see rate of contraction, and then eventually it will contract until minimum size because got no water inside already. Contractile vacuole then will shrink to minimum size. So that's what will happen at the very end. All right. And then the next question we already done just now, how the organism maintain its survival if the environment becomes dry. Basically, it does the same thing. It will form spores. So you can just write this exact same thing. So spores are very strong. So even though the surrounding is very hot, very dry, right, the spores will not die. So the spores will wait until, let's say, the temperature become normal again, then the amoeba will sort of like grow out from it. Okay. And then I think we are doing the last one already lah. Next page. Page 41. Now we do amoeba. Okay, amoeba, some of the process are quite similar. Some is a bit different. Now, number one, P is that the bulu on their surface. So you can see paramecium, a lot of hair one, right? So the hair called what first? The hair is called, yeah, you all still know this one, right? The cilia or even the cilium also can. Q is the, what's Q? It's not amoeba, don't have this thing, right? Cilium is singular, by the way. Cilia is plural. Both also can write one. They won't judge your grammar. So this is the vacuole, foot vacuole. Okay, this was not foot vacuole. This one is the contractile vacuole. So the contractile vacuole in paramecium will look different. They have two of it. They are star shaped. Okay, you can go to my Instagram. I go post one story about a paramecium undergoing osmoregulation. Yeah, you can look at that the actual video of how the contractile vacuole actually works. Okay, so moving on, they ask you to state the process involved. So same like the amoeba, it expel water because they live in the same place, fresh water pond. Water keep on going inside, they have to keep on expel water. So same osmoregulation. So that is the process of contractile vacuole. Please don't write excretion, must write osmoregulation. How P helps in feeding the organism. Now, for paramecium don't have what's that called? The phagocytosis. Don't have one. They actually have a mouth and a butt. The mouth is here. Look like a tongue shape like that. This is where food enters from. It's called the oral groove. This is their butthole, called the anal pore. And to eat food, right, they rely on this P. So how you going to explain this fella, right? This fella will like sweep the food. Move particle. Okay, there's a specific way to explain that. You cannot just say move the food particle. You must say they like this. So we will say as anus, anal pore, or actually your textbook will write anus, right? Yeah, anal pore or anus actually both also can. Yeah, both also can. Yeah, you must say it rhythmic beating or beat rhythmically also can. So you can say the structure P, which is your cilia, right? The full name. Beats rhythmically. So you must write that beats rhythmically. That's one mark. And then what does it do? And sweep foot particle into the mouth, which is called oral groove. So that is how we explain this. Can? Now, other than eating, right, cilia also involved in what? So when the cilia like that, it also help it to what? Help it to move, right? Yeah, movement, locomotion. So if they ask you how cilia helps in movement, right? Same. Beats rhythmically. Exactly the same thing we will write. Okay, beat rhythmically to help in movement. So that's another function. Movement and feeding both use the cilia. All right, the next one here, explain the life process that is shown here. Okay, you can see it's basically binary fission, right? One split into two, one. So you can say it is binary fission. The process or the live process. Like you said, the process is binary fission. Now this one 3 marks, they want you to explain a bit, right? Okay, the explanation actually is a bit chapter 6, like we learned recently in class. Okay, so mitosis. So basically you're going to add the word mitosis. So you just follow the words here. So I will say the parent cell will divide by mitosis to produce. So when they undergo binary fission, right, both of these will be exactly the same. That's why we learn mitosis, right? They produce what we call as genetically identical daughter cell. What's the difference between osmosis and osmoregulation? Okay, it's two different things. Osmosis is basically when water flows through a selectively permeable membrane from high water potential to low water potential. So that's osmosis, means it's just basically diffusion of water. Osmoregulation is whereby when living things expel excess water, like when you go to toilet and pee, that is called osmoregulation. Okay, so when they ask what is the function of contractile vacuum, you must say osmoregulation lah. Can so like osmosis basically is like just now when the water diffuse in that time, this is called osmosis or that's called diffusion water ma. But when they got too much water, they squeeze the water out. That's called osmoregulation. Chapter 3 mainly will have a lot of osmosis, but today we won't have time to do chapter 3 lah, right? And then remember for paramecium, right, when it's not favorable that time, okay, when they are favorable, also they will binary fission just like amoeba. But when not favorable that time, like if you look at next page, like this one, they say got change in pH in the pond, so we assume it's acid. They will carry out a type of sexual reproduction. This one is called conjugation, which is sexual reproduction. So for paramecium, not favorable that time, they will carry out conjugation. But favorable that time, same like amoeba, is binary fission. Okay, is binary fission and cell division different? Basically, binary fission is cell division. So basically, when amoeba want to reproduce by binary fission, it actually uses the concept of cell division, which is mitosis. But when they reproduce through this method, at time we use the word binary fission, but they are actually just dividing. But the method is called binary fission, but the concept is cell division mitosis. Okay. So today, I think I won't go forward anymore. It will be just until here. Actually, initially, I plan to do one more essay question for chapter 3.1, which is, if you have this with you right now, you can look at like this page 55. But I think it will take quite a lot of time lah. So this one, maybe I will try to do one video about it, or maybe I will do it in class also lah. Okay, so today will be just until here. I hope that all the questions I did help you to fully revise your Form 4 Chapter 2. So since there are a lot of people here that I never seen before, so if you have anything to ask, you can WhatsApp me. This is my number. Plus, please don't simply spam me like those hackers just now. If you got anything to ask about tuition classes also, you can ask me lah. All right. So if no questions, that is all for today. What is meant by kingdom for paramecium? Okay, that one actually is Form 5 only we learn lah. So that one you just ignore it lah. Yeah, that one only Form 5 we learn one. So that one is okay. Just ignore it. Okay, so if you don't have question, bye-bye. See you guys. Bye-bye, 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 bye-bye. Be able to access previous recordings. Alicia, you can WhatsApp me. I will answer you. I can send you some of it lah, but I cannot send you all lah. So the answer actually is no.